Hello, peoples.
Good afternoon, Dr. Slish. Well, I guess it's still morning.
Chat. Sydney's in. Hello, Sydney. Hello to everyone as well. As Sydney. Let's see, 1, 2, 4, 16 people here. Let's see, how does this thing work?
Are we going over the cell cycle today?
Yes, sir.
Okay.
We are cell cycling today. I'm just waiting a little bit because there were 2 different links for our meeting today. I don't know why. I think they're the same thing. But let me check up on that while I'm waiting for more people to come in. I just want to make sure they were both links to the same meeting. Otherwise, we're going to be missing people. Oh, I only have one now. So, let's hope they were the same. Bum, 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 bum. Bum, 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 bum. Okay. Watch, seller. Jelco. Okay, I guess we should just go ahead. So the beginning of this lecture, well, let's try and put it all in context a little bit. So in this chapter, we're going to talk about mostly what we talk about is signal transduction involved in controlling cell cycle. And that's really important because the next chapter is about cancer. And so cancer is a loss of control of the cell cycle. That's part of it anyway, a lot of it. So we're going to start out here with cell division. Now I'm going to brush over mitosis pretty quickly. There's some things you need to know about it, but kind of the processes of going on in mitosis is Bio 101, so I don't want to spend too much time on it. The signal transduction stuff is more complicated, and so I'd rather spend more time on that. Okay, so slide 2. Here's our cell cycle, which again, you should know this from Bio 102, but we've got, depending on how you count it, 2 phases or 5 phases or 9 phases or something, I don't know. But you've got the green here, which is interphase. An interphase is broken up into phases within interphase. Here's my chat window. Just feel like I should have. I'm sorry, I hate to keep stopping us, but trying to find where the chat stuff is. Oh, I can do polls. Life is good. Chat, there it is. Chat window. Okay. Can you record it and post it? Yes, I'm recording it right now. I recorded yesterday's too. I will post that. I should do that today. I think it says recording in the upper left. Yes, we are recording. So it uploads to my computer. So then I have to upload it to Moodle from there. So I'll do that after it does its thing when it's ready. Okay, Interphase. So interphase has got G1, S, and G2, which I'll talk about in a minute. And then metaphase, mitosis, which has got 4 phases in it as well. So we'll start out with G1. If you've got your notes there following along. G1 is like the cell is just divided into 2 cells, so now you've got a small cell. So in order to survive, the thing needs to grow up a little bit bigger. So G1 is kind of growth after cell division. And like most cells will be, there's no G0 here. Most cells will grow up until they're adult size and then skip out of the cell cycle and go in G0, which is called a rest phase. But they're not really resting, they're acting, they're performing their function. Most cells are in G0 because they're adults, they're doing what they're supposed to do, maintaining physiology in your body. But stem cells will always be in the cell cycle so they can keep on making more cells. So G1 cells, and we'll talk about passage from G0 back into G1 later. But once the cell gets up to adult size here and it's got all the signals in place, it goes into S phase. S is for synthesis. DNA synthesis happens here. The chromosomes get duplicated and the cell goes from 2N number of chromosomes to 4N. So for us, that would be 2 times 23 to 4 times 23. I don't do math, so you guys can do that. And then once the chromosomes are duplicated, you're into G2. And G2, I always say, is like, you know, you've got an adult-sized cell, and now you're going to make 2 cells out of it. So it has to get bigger and then duplicate all of its organelles and things so that when it splits into 2 cells, both cells have enough stuff to survive. So you've got growth before division and then growth after division to kind of get all cells the same size in the end. And then you go into mitosis, and we'll talk about the phases in a second. And after mitosis is cytokinesis. So phases of mitosis. So prophase, a lot of times people divide up into 2 different parts. Prophase, for me, is the most important one because we'll talk about why these things happen later when we activate a maturation promoting factor. Prophase, 3 things are going on here. The chromosomes are condensing. So during interphase, they're so diffuse you can't see individual chromosomes. But during prophase, they condense so you can actually see them as these X-shaped structures. The nuclear envelope dissociates into vesicles. And that's so that, you know, the chromosomes can be separated without that big membrane in the way. And the third thing that happens is the cell cytoskeleton falls apart, especially the microtubules. And they depolymerize, and during G2, the centrosome was duplicated into 2 centrosomes. So the microtubules depolymerize, and then they start to repolymerize in the form of the mitotic spindle. So those 3 things happen, and they're all controlled by phosphorylation events that we'll talk about later. So keep that in mind. Like in terms of mitosis, you probably know all this crap already, which is fine. Just remember these 3 things happen in prophase, which is really important. So you got your early prophase, you got your late prophase. Those 3 things are like in the process of happening from early to late. Now here in late prophase, you've got microtubules of the mitotic spindle attaching to the kinetochore. We'll talk about kinetochore later, but the kinetochore is a bunch of proteins attached to the centromere of the sister chromatids. So those sister chromatids have got like repeat sequences in the centromeres. So after division, after the replication, they're still kind of stuck together like this because of overlap with those repeat sequences that like the DNA opens up and then you get complementation between the 2 different strands. So they're kind of holding themselves together like that. And that's why you have that X shape. And all these proteins accumulate on there, and that makes up the kinetochore. So the kinetochore attaches to the mitotic spindle, and the mitotic spindle pulls those chromosomes back and forth until they meet in the middle somewhere, and that's metaphase. So this is like a dynamic tension phase. They're being pulled from either side, and because they're being pulled, they end up right in the middle. So you've got one sister chromatid facing one way, one facing the other way. And that then gets broken during anaphase. So here, anaphase, that connection breaks up. We'll talk about how that happens the next class probably. So that breaks up, and then the chromatids go in opposite directions. And another thing that's happening here in anaphase, sometimes they call this anaphase A and anaphase B. So at the very beginning, the first thing that happens is those chromosomes separate and start to move. And the second thing that happens is the cell elongates. So that mitotic spindle is pushing against itself and elongating the cell. And then you get to telophase, and telophase is basically just the opposite of prophase. So everything that happened in prophase unhappens in telophase. Chromosomes decondense, nuclear membranes form, and the mitotic spindle falls apart and forms that interphase cytoskeleton again. So that's mitosis. And I go over it fast because, like I say, you should know this from Bio 101. And I kind of hopefully pointed out some things that are a little more detailed than you're used to. Sorry about my burping there. That was very, very rude. Okay. And here's some pretty pictures. Okay. So at the end of mitosis, and keep in mind, mitosis isn't cell division. It's the division of the nucleus, division of the nuclear material and making two nuclei. The actual division of the cell is the cytokinesis. So cytokinesis depends on whether you are an animal or fungi or a plant. So the big difference here being cell walls. Animals and fungi don't have cell walls and plants do. So with your animals and fungi, what happens is a belt of actin myosin forms between the two cells, going right around the inside of the membrane between the two cells. And remember, actin and myosin interact during skeletal muscle contraction and a lot of other times. And when that happens, the belt tightens, 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 like that. So as the belt tightens down, the cells separate and you get two separate cells. Plants are a little bit different again. They always have to be different. What happens is you get vesicles with cell wall material and plasma membrane material kind of coalescing here. Come on in, honey. Say hello to the kids. It's my daughter, she's shy. So the vesicles fuse and then they form the wall between the two sides, much less dramatic. What's all that ding dong? Is it new people coming in or what? Oh, well.
Yes.
Yes, thank you. Okay, so that's cytokinesis. And so now I go into specifics in the notes about the mitotic spindle and different aspects of it. So you've got three different types of microtubules in this mitotic spindle. The ones that probably are most obvious, the ones that you think of in terms of the mitotic spindle, are the kinetochore microtubules. Let me just skip ahead a little bit. So here's the kinetochore. You've got all these proteins attached to the chromosome here, and they attach to the mitotic spindle. So here's your mitotic spindle, and they attach to these proteins of the kinetochore. And you've got a molecular motor in here, it's a dynein. It's going to go from plus to minus, so it's dynein. And it's going, you've got the motor there in the kinetochore, it grabs on the microtubule, and it's going to walk along the microtubule and pull itself towards the pole. So that's what your kinetochore microtubules are doing. And also on the other end, it's also being pulled from the centrosomal end. So you've got a dynein over here in the kinetochore pulling it this way, depolymerizing that kinetochore microtubule. And you also have a kinesin over here walking that way or pulling it from that direction and depolymerizing at that end. So it's actually shortening at both ends to pull that chromosome toward itself. Okay, so those are your kinetochore microtubules. Backing up, okay, so your kinetochore microtubules here. Then you've got these polar ones. They come from either side and they overlap. And in that overlap area, you've got molecular motors again, motor maps. I use both terms. I don't know which one you're familiar with. But these are going to be polymerizing, getting longer as the cell elongates. And you've got, again, some kinesins here, because kinesins are walking in the plus side. And as they walk to the plus side, they force those microtubules away from each other and force that cell to become more elongated. And then the last microtubule is the astral microtubule. See, I have a nice picture of this. Astral microtubules. Where are you? There. Okay. So here, the astral microtubules, you can see, are attached to the molecular motors that are on the, attached to the cortex of the cell. And those motors are pulling the microtubules towards themselves. So let me take a quick look at my notes. I may have messed that up. That, because it's walking from plus to minus, is going to be a dynein. Let me make sure I got that right. Oh, kinase is way too far. Mitosis, mitosis. Mitosis. Cytokinesis, microtubules. Yep. Okay, mistake in my notes. Dynein. Right? So that molecular motor then, that molecular motor is pulling, so it's basically walking from plus to minus and pulling that in. And what that action is going to do is, remember at the beginning of mitosis, your centrosomes are together here. And so they start out together and then they move down to the poles. So the astral microtubules are doing that. These centrosomes are shooting out microtubules to the poles, which then get grabbed by motors over there, and the motors pull them in. So that's what causes them to come down. So what's kind of interesting about this and why it's good that we do chapter 13 is in 13 we learned the generic stuff about microtubules, and you can see here how things moving around in cells are due to those microtubules and molecular motors. Okay, so those are our three different microtubules and molecular motors involved with them, and they're causing all of the movements we see during mitosis. Are there any questions about that? You can chat me or you can talk to me, or just give me a thumbs up or a thumbs down. Let's get a little feedback going here, people, because I went kind of fast. Oh. So I'm pretty sure you guys can interact with me over here. Thumbs up, thumbs up. All right, the only thing I didn't talk about just now as I was going through really fast is going back to my notes. So up here in prophase, I throw in some targets of phosphorylation. So here's one to keep in mind. You'll see them talk about this in one of the animations I loaded up for you. Nuclear lamins. So nuclear lamins are holding the nuclear membrane together. So when you activate mitosis, a kinase comes in, phosphorylates those lamins, and when they're phosphorylated, they depolymerize. And the lamins are kind of like the cortex of the cell. They're holding the plasma membrane in that place. Here, the lamins are holding the nuclear membrane in place. So when they depolymerize, the phospholipid bilayers of the nuclear membrane become vesicles. So that's an important phosphorylation target. Here's another protein called condensin. Love it when a protein tells you what it's doing. So just get rid of that red line. So condensin then is going to be involved in condensing those chromosome. So when that gets phosphorylated, it gets activated. And cytoskeleton, yeah, I don't have any specific target here, but remember those two because we'll talk about them later. Cytokinesis, microtubules. Okay, I'm going to move on and get to the more, so kinetochore microtubules, polar microtubules, dynein, astrokinesin. Well, let's take a look at it. You know, I tend not to like memorize things in that way. Whenever I try it, because it, you know, when you've got a 50-50 chance of getting it right, oh, look at my hair. When you got a 50-50 chance of getting it right, you got a 50-50 chance of getting it wrong. So whenever I look at this, when I think about what's the motor, I think, well, where is it moving in relation to that centrosome? So here on the kinetochore, it's moving from plus to minus, so that's dynein. On the centrosome, you've got a motor that's walking from minus to plus, so you've got a kinesin there that's pulling that microtubule in. Over here on the polar, you've got kinesin because it's going to be walking from minus to plus, right? It's walking in this direction as it's forcing that microtubule back. Astral over here is again walking from plus to minus, so that would be dynein. So, you know, it depends on, you know, I always look at it mentally back in the cell so I can determine which is which, because I'm old, my hard disk is full, I can't really memorize stuff as well. Okay, any other questions about microtubules before we move on? No? How's everyone getting along at home? Have you gotten any arguments with your father, your mother, your parents, siblings? I do nothing but fight around the house. Gives me energy. No, I'm just kidding. Okay, moving on. Control of the cell cycle, checkpoints. Let me bring my notes up so I can just remember how I talk about this. Okay, so here are your different phases. You've got 3 different checkpoints. Now this checkpoint here from G1 into S, and this one over here from G2 into M, those 2 checkpoints are controlled by similar mechanisms. And there are other kind of lesser checkpoints through the middle that are controlled by those mechanisms as well. But those 2 are very similar. This third one up here is very different. This is kind of that pause in metaphase before the cell actually splits those chromosomes. So this one we'll talk about separately from these 2, which are very similar. And in terms of these 2, most of the early work was done on these, on this one rather than this one, just because it was easier to get to. And there's a really nice animation that I put into in here, I believe. Where is it? Cycle control. Oh, okay. Discovery of control proteins. So watch that. It talks about what I'm going to talk about in the next couple of slides. So basically, we've got these 2 checkpoints. Now this one is really important in that once the cell gets past this checkpoint, it is committed to divide, because once it gets past the checkpoint, it immediately starts replicating its DNA, and a cell can't really survive with more than the normal number of chromosomes. So it commits the cell. So that was a really important control point. Also, if you lose control of that checkpoint, the cell just keeps dividing and dividing and dividing, which is bad. You know, that's where your cancers come from. So this is our most important checkpoint. So this is really the control over cell division. This one over here, the G2, is just making sure that the cell is ready to go through mitosis. If you've got DNA damage, or the chromosomes aren't finished replicating, or the cell's not big enough, it's going to block the cell from going through mitosis until it's ready. So that's what that one's all about. So this is, yeah. So most of your cells are going to be in G0, and most are post-mitotic. They will never divide. Like your nerve cells are never going to divide. Muscle cells, for the most part, don't divide. Other cells like liver cells, if you have some liver damage, they can be induced to divide. You can make new liver cells. So those are cells that are in G0, but can be stimulated to divide again, to go back into that cell cycle. And then you've got other cells like stem cells that are constantly being stimulated to go through, to be in the cell cycle. And they're making more and more cells all the time. That's their whole purpose. So this site here, this restriction site from G1 to G0 is going to be controlled by growth factors. So we talked about receptor tyrosine kinase mechanisms in the last chapter, and TGF beta-like mechanisms, the Wnt pathway, all of those things are important in controlling this checkpoint. So normally if a cell is not being stimulated by that, it will not divide. Then you've got these other things here on the slide, nutrients, cell size, cell damage, which is also important, especially in terms of DNA damage. We'll talk about that later with P53. If the cell has got DNA damage, P53 gets activated, and that blocks cell division no matter what. Okay, so, all right, so how is this discovered? Let's, this is your G1, side note, yes, I've got those. We'll be good. So one of these early experiments they did was they took some cells that were in S phase, so they're already past that G1 point, and they took some that were stuck in the G1 point, fused them together, and the one that was in S phase forced the other one to go in S phase. So it tells you there's some kind of a signal that's been activated in the S1 that causes that getting through that pathway. Same thing here with, this is Xenopus oocytes again. So your oocytes, they go through one round of meiosis, and you get a polar body and an egg, and then they kind of freeze in G2 before that second round of meiosis. And when the sperm activates the cell, it goes through that second phase of meiosis to become, and then you've got three polar bodies, something like that. You get two polar bodies and then one egg cell. But anyway, that last kind of meiosis round, it's frozen right before that. So to get the egg to become mature, the sperm comes in and that matures the egg, and then it's off to the races. So what they did in these experiments was they took an egg cell that was already induced to go into that, and then fused it with one that wasn't there, and they see something that was in the mitosis one activated the stopped one to go through mitosis. So something in the cytoplasm is activating these cells. This is more interesting and more important. So now they want to figure out what is it in that cytoplasm that's activating the cell. So they suck some cytoplasm out of an activated cell, they put it in a frozen cell, and that activates the frozen cell. Great. Now we've got cytoplasm. We've got something we can work with. They take the cytoplasm, they break it up into proteins. There's tons and tons of proteins in the cytoplasm, and they check them to see which proteins are the ones that are important to cause this maturation process. And when they found out what the proteins were, they called it maturation promotion factor. Okay, maturation promoting factor. So again, this is like this is before they did a whole lot of DNA work. This is probably 1970s, so there wasn't a lot of DNA work being done yet. So still they don't know what those proteins are. They're just calling them something that causes maturation. All right, then you take those and you take them apart and we figure out what actually is causing maturation. And it comes down to two main proteins. You've got CDK, which is a cyclin-dependent kinase. It is a kinase, so it's what's doing the phosphorylations that are causing things to happen. And it's cyclin dependent. It's being activated by this control protein cyclin. And cyclins are called cyclins because they go up and down during the cell cycle on a regular kind of clock pathway. So certain cyclins come up right before the G2 to M transition, and then they go down. And then other cyclins will come up before the G1 to S transition, and then they go down after it. So they cycle through the path, through the cell cycle, and that's why they're called cyclins. But anyway, here with maturation promoting complex, you get the cyclins coming together, and then there's some phosphorylation events that happen, we'll talk about later, that fully activate the complex. But once it's fully activated, that cyclin and CDK together are called MPF. That's what MPF was. It was two proteins, not one. And it's when it's active, it's called MPF. Now your CDK then will phosphorylate specific proteins that will turn things on, like condensin. Phosphorylates that, causes it to condense the chromosomes. It'll phosphorylate the lamins, cause the nuclear membrane to fall apart. And then eventually it will also activate some, what are they called? Ubiquitolating enzymes. And they will then ubiquitolate cyclin, and then cyclin gets degraded. So MPF kind of turns on the transition into mitosis, and then it turns itself off by activating enzymes that phosphorylate, that ubiquitolate it, and then it shuts itself off so the cell can come out of mitosis. So I think we're going to talk about that more later, but at this point, mainly what we're interested in is you got your cyclins, you got your CDKs. And there are different cyclins and CDKs depending on what part of the cell cycle you're in. So if we go back to the notes, another nice animation there for you to look at. By the way, there's tons of animations. Some I like more than others, but try to focus on just what I'm talking about. So do some YouTube searches if this is unclear, because there's tons of stuff out there. Okay, so in terms of CDKs, we've got really 4 that are important. CDK1 usually will be called CDC2. Remember CDC42, CDC this, CDC that, those are all cell cycle division something is what the CDC stands for. So these are all proteins that before they knew what they were, they're like, here's protein 1, 2, 3, 4, up to 42 of the different proteins that are important in cell cycle control. So CDC1, it turned out in retrospect to be a kinase, and it is CDK1. So CDK1, CDK2, same thing. Then you've got CDC2. Then you've got CDK2, which is going to be important in getting through that restriction point, that G1 to S transition. And we've got CDKs 4 and 6, which get you up to that transition point. So we'll talk about that a lot more later, but you've got 3 different, usually we say, you've probably seen this before, 4/6, which means really both of them. Okay, moving on. Then you got your cyclins, you got your cyclins, you got your cyclins. So let me throw in the cyclins here. You've got isoforms of these, isoforms, which are cyclin, where do we start? Let's start with D. Oops, not that. E. So cyclins D and E. D is going to get you up to the restriction point. E is going to get you through it. When you're in S phase, cyclin A becomes important. As you come out of S phase, then cyclin B is the one that's involved in MPF. It's going to bind to CDC2 and get you through that transition point. So we've got really 3 different CDKs and 4 different cyclins that are important. And I think I said this already, cyclins activate CDKs when their concentration peaks. Oh, I didn't say that. Let's look at this. So let's look at activation of MPF. So this blue line here is the concentration of cyclin B in the cell. So, and then the black line is activation of MPF. So the black line is when CDC2 and cyclin B get together, then they're active MPF. And there are other phosphorylation events we'll talk about next time. But that is MPF. So you can see here that the cyclin level increases slowly throughout the cycle until you get past some certain threshold value of cyclin B concentration. And when you get past that value, MPF starts up. So boom, 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 you get activation of MPF, MPF peaks, and that peak of MPF is getting you through the metaphase transition, through the G2 to mitosis transition. When you peak there, then cyclin starts to get degraded and it comes down. And as cyclin is degraded, MPF activity shuts off. And as soon as it's degraded, it starts building up again. So it's kind of interesting in that cyclins are rare among proteins, cyclin B especially, in that it's being controlled by its degradation, not its synthesis. Synthesis is pretty much constant, but once it peaks, it gets broken down and then has to build up again. Kind of like beta-catenin, right? That's being controlled by its degradation as well. And here's my assistant, Gus. Hi. He doesn't know as much about this as I do, though, so direct your questions to me, not him. All right, that's, I think, about all I plan to talk about today. Oh, God. Not Facebook notifications. Okay, so anyway, I don't think I wrote this in the notes, but we can talk about them. I'll add it to the notes later. Here we are looking at the cell cycle. So you've got your mitotic CDK, which is CDC2 or CDK1, and it's kind of in the cell inactive through most of the cell cycle until the mitotic cyclin builds up high enough. Then it, get out of here. Then it binds together, forms MPF, and MPF does this, causes nuclear envelope breakdown by phosphorylating those lamins, chromosome condensation by phosphorylating condensin, mitotic spindle formation by activating non-motor maps, and they're controlling the catastrophe and then repolymerization of the microtubules. And all these things are what I was saying earlier were prophase. So MPF directly causes prophase by doing that. And it also activates some targeted protein degradation. Among one of the things it targets to degrade is its own cyclin. So it degrades cyclin B in the process of activating mitosis, and then that allows the cell to get out of mitosis in the end. And it's also going to help with the M to A transition that we'll talk about later. But targeted ubiquitination is going to be activated by MPF as well, and that will be involved in this M to A transition, metaphase to anaphase. Okay, preguntas, mi gente? Any questions? Why, Dr. Slish, did you not shave when you knew you had class? I don't know. My wife kind of likes it. No, she doesn't. She hates it. No questions. Nobody's chatting me. Nobody's talking. I'm feeling kind of alone now. Can I get a little feedback? Can I get a little love? How are we doing here? That's about it for my lecture, which is done in 42 minutes. Raphael gave me a thumbs up. Give me a thumbs up if you're good, or not if you're not good. Thumbs up, thumbs up. Thumbs me, people. Sydney. Oh, I got claps from Perez. Nice. How about Jana? Jana. It's funny. Some of you, I can't picture your faces because a lot of you I do know. Some of you I don't. Emmy's good. Like the Zoom meetings more than chat's good. I'm good. Do you like it more than chats because of my typing? My typing's bad. Caitlin Bajelko. How we doing there, Caitlin? Jenny Chen? Ariana? Everybody good? Quach? Quach is always good. You ask good questions, Daniel. Keep it up. Okay. Are we good? We're going to call it a day? Most people say good. Rachel says good. All right, well, why don't we call it a day then? And we can all get back to stress eating and gaining weight.
Have a good day.
Okay, you have a good day too. Stopping video recording and ending meeting. Bye, guys. One last comment. Yes, I am posting these videos to Moodle.